So I'll jump in first with a follow on to what I was just talking about, and that is, phage display and now understanding the power of CRISPR and how bacteria use it, and actually now turning that for phages to use against bacteria, in terms of phages being able to essentially gene edit bacteria and kill them is an incredibly intriguing idea. Again, these kind of biologics have had a problem advancing to the clinic, so we'll yet to see, but that's very interesting. I think, from the natural product side, this is also a really rich time where we get to go back and look at old natural products whose mechanism of action was not really understood, but at the time maybe it didn't meet the bar for kind of developing or studying further. But now, because of the predicament and we're in, we actually can go back and understand new science around old natural products, but also modifying organisms as well as changing kind of biosynthetic pathways to create new secondary metabolites. I think we're in an era now where the richness of natural product discovery for antibiotics is going to be really interesting. But I think for me personally, probably the most exciting kind of frontier of antibiotic discovery is thinking about biofilms. And so, biofilms have been around for a while, as microbiologists have known about them for 30 years, but they really haven't made their way into kind of the detailed study by, for lack of a better term, kind of molecular scientists or people like chemists that are really thinking about these things at the molecular level. And I think as we get to understand biofilms and how bacteria actually live in infections and in communities, there's going to be a lot of interesting strategies that come out about how to control infections, right? Because it's not about keeping no bacteria in your body to stop an infection. It's about not letting too many bacteria get out of control in your body and your immune system not being able to keep up. So, a lot of that, stems from chronic infections and biofilms, and we're just scratching the surface of what those communities look like in infectious disease.
Yeah, well put, actually. The only thing I can add to that is that there's also the field of organic chemistry and organic synthesis has matured to a point where what would have been natural products and their structures would have been challenging to access by chemical methods. Today, not only are those chemical methods in place, but one can design approaches that allow structural modification within the backbone and within any site within the molecule. And so it's really a wonderful time for chemists to actually use their talents to create non-natural antibiotics that match or exceed the natural products themselves.
Yeah, and that perfectly said. And as you know, we both believe in the power of us to be able to do that. I'll also add to that I, as a young chemist that has gotten just absolutely fell in love with antibiotic discovery, have really appreciated the difficulty of antibiotic discovery in the sense that, you know, if we think about medicinal chemistry, we might think about, okay, we have a biochemical target, and therefore we have to develop a structure activity relationship for this molecule. But for antibiotics, there's a structure activity relationship for the biochemical target. There's a structure activity relationship for entrance into the bacteria. There's a structure activity relationship for efflux out of the bacteria before you even get to kind of the medicinal chemistry and pharmacology side of things. And so even if I think at that basic level, if I have to take 3 levels of SAR and overlap them to get a clinically or a therapeutically relevant compound, that's really challenging. And so we're finding that, the differences, the ability to navigate between those SARs is really an a single atomic change to these molecules. It's one methyl group. And so our synthetic ability now, it's one fluorine group to be able to modulate a pKa. Our synthetic ability to go in and really change molecules, complex molecules in that fashion now is really exciting. So I agree, Dale.
It's an exquisite issue. I mean, there's giants in the field that are contributing articles that we feel really lucky to have brought together in a single issue. And so if someone wants to gain an appreciation of where the field stands and what some of the giants in the field are doing, directions that they're taking, initiatives that they've taken on. It's a great, not just introduction, but deep dive into where the field stands at this stage and where some of the challenges are and what some of the approaches are being taken to solve those challenges.
I agree. It's an amazing compilation of articles and scientists that are trying to address this. And that's for scientists picking it up. If I think about students picking up this issue, I think it's incredibly important for them to pick it up and appreciate not only the strategies but the type of chemistry, and start appreciating the differences in kind of the antimicrobial chemistry, biology, and medicine space versus other spaces. Because going back to some of your previous questions, that's, to me, a very big concern as an educator as well, is that without the industrial kind of contributions in this field, we're actually losing a lot of expertise in this space. And it may sound trivial, but I think it's true that the infectious disease space from a medicinal chemistry standpoint is very different from other spaces. And maintaining education, students, and people that have expertise in this area is very important for us as a society.